Very good. Looks like we're getting up there to the vicinity of our usual numbers. I want to make sure we started a minute late. I want to make sure we're respectful of your time. So I'm actually going to start introducing you now as people continue to come in. Well, welcome everyone. Today we're quite lucky to have Hunter Shain as our global dermatology talk speaker. Dr. Shain completed his undergraduate training at the University of Illinois. He subsequently completed his PhD training at Stanford University, where he worked with Jonathan Pollock and worked on pancreatic cancer genetics. He did his postdoc at UCSF, where he worked with Boris Bastian, and that's what kind of pulled him closer to derm, so to speak, where he worked on melanoma and melanocytic nevi genetics. He's now the assistant professor in the Department of Dermatology at UCSF. Dr. Shain's an expert in the genetics of melanocytic tumors. His work for me has been really fun to follow over the years. He's published some of the most seminal studies in the past 5 years or so in the genetics of melanoma, melanocytic nevi, and most recently normal melanocytes in skin. And so he's published extensively, including in Nature, a New England Journal paper a number of years back that I've personally read probably 5 times as a reference. And he's won numerous awards for his work. So we're really excited to have Hunter with us today, and take it away.
Great, thank you. I'm going to start by sharing my screen, and I will start the slideshow. Okay, so I hope you guys can hear. Just chime in, or sorry, hear and see. Chime in if you cannot. I wanted to start out by thanking the organizers for the opportunity to speak today. I think this is great that you've organized these global dermatology talks. And today I'm going to talk to you about the genomic landscape and evolution of skin cancer. And I'm being purposely vague in the usage of the term skin cancer, and that's because in today's talk, I want to cover both melanoma and cutaneous squamous cell carcinoma. We'll start with melanoma, and I always like to remind everyone that melanomas are clinically heterogeneous. So the 2 most common forms of cutaneous melanoma are the low and high cumulative sun damage subtypes of melanoma. High CSD melanomas arise on body sites that are chronically sun exposed. So this is typically going to be your head and neck regions. They increase in incidence with age, have extraordinarily high mutation burdens, and a unique set of driver mutations that I will cover in more detail in the following slide. By contrast, the low cumulative sun damage subtype of melanoma, these tend to arise on body sites that are more intermittently sun exposed. So this would be your back, your shoulders, your chest, and your trunk regions. They have a somewhat peculiar age of incidence. They seem to increase in incidence until around the 5th decade of life and then decline thereafter. Now, compared to completely unrelated cancers, let's say, breast or prostate cancer, their mutation burdens are certainly high, but compared to the low or the high CSD form of melanoma, their mutation burdens are somewhat lower, and they too have a unique set of driver mutations that I'll cover in more detail in the next slide. Now, these concepts are often confusing to scientists, and that's in part because these types of melanoma have gone by different names throughout the years. So when you read older publications, the high CSD form of melanoma is often referred to as lentigo malignant melanoma, and the low CSD form of melanoma is often referred to as superficial spreading melanoma or non-CSD melanoma. And in fact, when we wrote this review in 2016, non-CSD was the terminology being used, which is why it appears in this figure. But today, the World Health Organization recommends the adoption of the terms low and high CSD forms of melanoma. So as I mentioned, these 2 types of melanoma have distinct sets of driver mutations, and this is my best attempt to collate all of the genes with bona fide pathogenic mutations in the various subtypes of melanocytic neoplasia. Now, there are quite a few genes listed here, but they are not necessarily all implicated in every subtype of melanoma. And so I'm highlighting here in the darker shades of red or blue, those genes that tend to be mutated in roughly 10% or more of a given subtype of melanoma. So we'll start with BRAF. Low CSD melanomas have a high frequency of BRAF V600E mutations, which results in activation of the MAP kinase signaling cascade. A high frequency of biallelic loss of function P10 alterations, which results in activation of the PI3 kinase signaling cascade. Frequent TERT promoter mutations, which upregulates telomerase and thereby immortalizes those cells. Frequent biallelic loss of function mutations affecting the CDK into a tumor suppressor gene, the main endpoint being to disrupt cell cycle checkpoint control. Frequent TP53 mutations, which perturbs the P53 pathway, and then loss of function mutations affecting the ARID2 tumor suppressor gene, disrupting a critical component of the switch-SNF chromatin remodeling complex. Now, by contrast, high CSD melanomas, they actually share many of these complementary mutations in other pathways, but they have distinct mutations predicted to activate the MAP kinase signaling cascade, including a high frequency of NRAS mutations, primarily affecting codon 61, as well as a high frequency of loss of function NF1 mutations, which operates upstream of NRAS. Now, to be sure, there have been a long tail of many diverse types of MAP kinase pathway activating alterations implicated in high CSD melanomas, but again, I'm just trying to show you those that seem to occur in roughly 10% or more of a given subtype. So I now want to pivot, and instead of discussing the different types of melanoma, let's discuss how they evolve. And I had the pleasure of delineating the genetic evolution of melanomas with Boris Bastian. And in those studies, we took advantage of particularly informative cases in which we could identify a melanoma at that moment, in which it was growing out of its remnant, but still intact precursor neoplasm, such as in the example shown here. So what we did was we microdissected and sequenced the surrounding normal skin, which simply served as a genetic reference or comparison. And then we also microdissected and sequenced the precursor and descendant portions from these combined neoplasms, allowing us to infer the order in which genetic alterations and gene expression changes occurred during their evolution. To severely oversimplify these studies, a common progression trajectory emerged, and that was for a melanocyte to acquire a BRAF V600E mutation, resulting in the formation of a common nevus, and then for a cell within this nevus to acquire mutations in other complementary pathways, resulting in its transformation into a melanoma. Now, we felt like these were informative studies, but there were still some major questions that remained unanswered. And perhaps one of the biggest questions, and in fact, there were editorials written to the New England Journal of Medicine asking about this, was where do melanomas that arise de novo come from? So when you talk to patients and their care providers, they will swear to you that in many cases, if not most cases, the melanoma just appears out of nowhere, meaning that there's no obvious precursor lesion anywhere to be found in the vicinity. If you talk to dermatopathologists, they would estimate that about 1/3 of melanomas arise from common nevi. So here we hypothesize that maybe these mutations could occur in reverse order. So for instance, let's say a melanocyte first acquires mutations in these other pathways that are thought to eliminate secondary and tertiary barriers to tumorigenesis. Well, that might not result in a discernible phenotype, but if that melanocyte subsequently acquires a BRAF V600E mutation, the ensuing neoplasm could skip the precursor phases and manifest immediately as a melanoma. Now, another major question facing the field was, where do the high CSD melanomas come from? So as you will remember from the previous slides, high CSD melanomas are notable because they do not have BRAF V600E mutations. And yet when you sequence common nevi, the vast majority do have BRAF V600E mutations. So it seems like we've got this whole half of melanomas that are missing a set of precursor neoplasms, and yet they must be coming from somewhere, right? So here we hypothesize that maybe these mutations are occurring with some frequency, but they're too weak to result in the formation of a discernible neoplasm. Nevertheless, when eventually combined with mutations in other pathways, they too can transform into melanoma. And I want to mention that the mutations observed in high CSD melanomas, there is biochemical evidence to suggest that these are more weakly activating at the pathway level than the BRAF V600E mutations. Now, it's always fun to lay down hypotheses, but then comes the challenging part. How do you test them? And in this case, we felt like the most straightforward way to test this would be to genotype individual melanocytes from physiologically normal skin, and then ask the question, do these melanocytes ever harbor pre-existing pathogenic mutations? Now, this is not straightforward, and the reason is that melanocytes from normal human skin are polyclonal, meaning that each melanocyte from our normal skin has a distinct set of somatic alterations. and it remains difficult to this day to genotype an individual cell at a high degree of specificity and sensitivity. And so to overcome this rate-limiting step, we have optimized a tissue culture protocol to reliably induce a limited number of population doublings from single-cell sorted primary human melanocytes. Now, if any of you have ever attempted to culture primary human melanocytes, you know that they do not like to be split thin. And of course, you cannot split them any thinner than single-cell sorting them. So I don't want to pretend like we can take one cell and happily expand it to fill a 10-centimeter dish with millions of cells. But we have found the right conditions to reliably eke out a handful of population doublings. And believe me, it is much easier to genotype from a starting point of 2, 4, 8, 16 cells than it is to literally genotype from one cell. Now, the second experimental solution we've invoked is we have established in our laboratory what is called the GNT-seq protocol, which outlines how to extract, separate, amplify, and sequence both genomic DNA and mRNA from an individual cell, or in this case, a small clone of cells. Now, there are many advantages to having both DNA and RNA sequencing information from the same cell, but one of the main advantages is that it provides an internal benchmark by which to assess the quality of your somatic mutation calls. Now, I'm going to gloss over quite a few bioinformatic details that ultimately went into our genotyping workflow. The citation information is below if you want to read about them. But the bottom line is that we can call somatic mutations at nearly 100% sensitivity and specificity from melanocytes from human skin. So what have we genotyped thus far? Well, we've currently genotyped 253 individual skin cells. The vast majority of these are melanocytes, which will be the sole topic of today's talk. These cells come from 31 donors and 45 skin biopsies. So we were fortunate to have the ability to biopsy some donors multiple times, which is why there's not a one-to-one ratio between donors and biopsies. I'll mention we did publish a portion of this data last October, but our current sample size is nearly double what has been published. And I'm happy to overview all of this data today. Let's begin by overviewing the mutation burden of individual melanocytes from human skin. In this plot, each data point corresponds to the mutation burden of an individual cell. These melanocytes are grouped together based on the biopsies from which they arose. And then these blue lines correspond to the median mutation burden of individual melanocytes from a given skin biopsy. So as you could see, these skin biopsies display a wide range of mutation burdens. And so we wanted to ask what features primarily explain this variability. So to begin, the skin biopsies with the first and third highest mutation burdens were notable because they were peritumoral skin samples, meaning that this is the normal skin, but the skin adjacent to melanoma. And I really want to emphasize that this is normal skin. This is not an extension of the melanoma into that area, but because of its proximity to the melanoma, we believe this is why these skin samples have such a high mutation burden. Now, the sample with the second highest mutation burden was notable because of its extraordinary degree of freckling. We had these grand ambitions that we would microdissect out individual freckles to better understand their genetic basis. Didn't quite work out as planned, but what I can comment on is that the mutation burden of individual cells from this larger piece of skin were quite high. Moving on, skin samples from patients with melanoma have high mutation burdens. This would, of course, include the peritumoral skin samples that I showed you 2 slides ago, but this also includes skin samples from a patient with history of melanoma, but the skin biopsy here was not necessarily adjacent to the melanoma. So let's say, for instance, you have a history of melanoma on your forehead, and then this biopsy comes from your lower back, then you would be colored in this tiling plot. Moving on, skin samples from patients with a history of tanning bed usage have high mutation burdens. This is especially impressive when you consider the fact that all of our tanning bed donors were quite young. They were in their late 20s, 30s, early 40s. And they're actually being compared here to a non-tanning bed cohort that is nearly twice their age. So most of these donors are in their 70s or 80s. And we're currently working to get a better control cohort. But even without that, you can clearly see the relationship here. And I'm going to circle back to this point in a couple of slides. Skin samples from patients with dark skin tone tended to have few mutations. So in this tiling plot, the white tiles correspond to those donors who identify as being of African ancestry. And then the gray tiles correspond to those donors who identify as being Hispanic or of Asian ancestry. And generally speaking, you can see that their mutation burdens are somewhat lower. Next, this is a finding that continues to surprise me, but we've observed it enough times that I do not believe we can dismiss this as a series of outliers. And that is that skin samples from intermittently sun-exposed skin tended to have more mutations than skin samples from chronically sun-exposed body sites. So in this tiling plot, the black tiles correspond to biopsies that come from the back, the shoulders, the chest, or the trunk regions. These are classically body sites that are categorized as intermittently sun-exposed. The gray tiles correspond to biopsies that come from the head and neck region. These are classically body sites that are categorized as chronically sun-exposed. And then we did sequence a small number of sun-shielded body sites, so either the buttocks or the foot sole, and not surprisingly, these skin samples had few mutations. But why are the intermittently sun-exposed biopsies displaying such high mutation burdens? Well, to begin, I'm not convinced this is necessarily going to hold true for everyone. So thus far, all of these donors had occupations that I would, consider to be white collar occupations. So this is your, office workers. I suspect that if we genotype head and neck melanocytes from farmers, construction workers, anyone who works outdoors for a living, that this relationship might not hold. Nevertheless, it seems a bit counterintuitive. That is, until you consider the anatomic distribution of melanoma. So most people don't know this, but melanoma is actually most common on the backs and shoulders of men, and the trunk, particularly the thighs, of women. Now, some have argued that this simply reflects the fact that those body sites have more surface area than our head and neck region. So obviously they should have more melanoma. But even when you mathematically account for surface area, it seems like melanomas are occurring more often on our intermittently sun-exposed body sites than they should, especially if you compare their anatomic distribution to that of keratinocyte cancers, such as squamous cell carcinoma and basal cell carcinoma, which show an unequivocal preference for the head and neck regions. So why might this be happening? Well, we currently hypothesize that maybe melanocytes from different body sites are biologically distinct. And I think this is bearing out both in cell atlas studies as well as model organisms, such as recent work in zebrafish from Richard White's lab. We believe that there would have been very strong evolutionary selection for melanocytes on chronically sun-exposed skin to not transform prior to the end of reproductive age. And as such, it is tempting to assume that maybe these melanocytes have evolved better DNA repair capacities to withstand the chronic onslaught of UV radiation-induced damage. So this is a hypothesis that we look forward to testing as we move forward. Moving on, there was a poor correlation between age and mutation burden in this data set, and I'm not prepared to stand here and claim that as we get older, the mutation burden of our melanocytes stays the same or somehow even reduces. But what I think this reflects is the fact that other variables, such as your personal exposures to UV radiation, are simply more important. And in particular, in this data set, all of our, or most of our younger donors, those who were less than 40 years of age, were from our tanning bed cohort, which probably skewed their mutation burdens upwards to an extent. Now, I have presented a lot of information, and some of these features do counteract one another. So I wanted to take this opportunity to summarize in a single slide the variables that we believe are most important in dictating the accumulation of mutations in melanocytes from human skin. So a previous history of melanoma, freckling tendency, and tanning bed usage seem to be highly correlated with mutation burden. This is followed by one's skin tone and anatomic site, where somewhat counterintuitively, low CSD or intermittently sun-exposed body sites seem to accumulate mutations more rapidly than high CSD or chronically sun-exposed body sites. And then finally, I do think that age is important, but it is less important than one's personal exposures to UV radiation, as well as constitutional factors such as your skin tone. Now, I find all of this interesting from a purely basic science standpoint, but it is worth asking whether this can be clinically useful. And I think the answer is yes. So melanoma is, in theory, one of the most preventable types of cancer. Prevention strategies generally fall into two main categories. There are primary prevention tactics, which means to intervene before health effects occur. So this would be the usage of sunscreen and the adoption of sun avoidance strategies. And then there are secondary prevention tactics, which means to screen in order to identify diseases in their earliest stages. So this would be a routine skin examination by a healthcare professional. And so it is worth asking, given that we have these tools at our disposal, why does anyone get and die from melanoma? And the unfortunate reality is that our medical system cannot screen everyone all the time. We just cannot absorb that burden. And there are just no clear guidelines on who should be screened for skin cancer, when should they begin screening, and how aggressively should we be adopting primary prevention measures. I mean, you can compare the prevention guidelines to, let's say, breast cancer, where there is a very clear age if you do not have a family history of breast cancer, at which time women are recommended to begin getting mammograms. But what age do you start getting screened for skin cancer? If you have dark skin tone and you avoid the sun, maybe never. If you have light skin tone and you go to the tanning bed, maybe you start getting screened in your teens. And so I want to pitch to you my futuristic vision. And that is that we could use my assay, or ideally a higher throughput variant of it, to measure the mutation burden of skin cells at various points in one's life. So this cuts through all of those challenges of inferring damage, and you just directly measure the accumulated damage. And if you have an accumulated level of damage that falls outside of the normal range, then you would be a candidate for enhanced primary and secondary prevention measures. So this is a goal I look forward to hopefully realizing one day. Now I want to pivot, and instead of discussing the number of mutations from melanocytes in normal human skin, let's discuss the types of mutations. And one finding that was stunning to us was that pathogenic mutations, meaning melanoma-causing mutations, were ubiquitous in melanocytes from so-called normal human skin. So this table summarizes all of the instances of pathogenic mutations that we observed. By far the most common were loss of function mutations affecting the NF1 tumor suppressor gene, followed by loss of function mutations affecting the RA2 tumor suppressor gene, then gain of function or hotspot mutations affecting the BRAF oncogene. But interestingly, what was missing, we did not find any BRAF V600E mutations. So most of these are more weakly activating mutations that affect other codons. And then finally, I won't read off the rest of these, but you can see them listed here. We wanted to get an idea as to how frequently were these pathogenic mutations occurring. And so we subdivided the skin samples from the previous slide into those with the highest, lowest, and in-between mutation burdens. And as you can see, generally speaking, those with a medium or low mutation burden tended to have relatively infrequent pathogenic mutations. But 35% of melanocytes from the skin biopsies with the highest mutation burdens harbored a pathogenic mutation. And I want you to take a moment to internalize this. Think about the person you know who has the most sun-damaged skin, and the notion that 1/3 of their melanocytes harbors a melanoma-causing mutation in their sun-exposed skin cells. It's stunning that our body, or the cells in our body, have developed tumor suppressive mechanisms to prevent them from transforming. But on the other hand, when such a high frequency of poised potential precursors exists, it seems like it's just a matter of time before one of those is eventually going to transform into melanoma. Now, circulating back to the types of mutations, you will notice that the majority, not all, but the majority of these mutations are predicted to activate the MAP kinase signaling cascade. And indeed, these are highly overlapping with the types of mutations that are seen in high CSD melanomas. And so we were wondering if these mutations were inducing a proliferative phenotype. And indeed, we found evidence that they were. So this is just one example. We found many other examples, but in this skin biopsy, we sampled a piece of skin that measures roughly 7 millimeters on each side. We genotyped 15 individual cells from this area of skin. Nearly half of these melanocytes had a mutation predicted to activate the MAP kinase signaling cascade. And interestingly, several of these cells were phylogenetically related, meaning they share some, but not all of their mutations. Now, the most parsimonious explanation for this finding is that there was a parental cell, which acquires a mutation in the MAP kinase signaling cascade that then caused it to give rise to a field of daughter cells. And then by sequencing multiple cells within that field, they would, of course, share the mutations inherited from their parental cell. So that formulates the trunk of the phylogenetic tree. And then they have private mutations on the branches of the phylogenetic tree that accumulated since the most recent clonal expansion. So given this discovery, and in conjunction with some of our previous work, as well as work by others, we now propose that there are 3 routes to melanoma. The first route is for a melanocyte to acquire a strongly activating mutation in the MAP kinase signaling cascade. This is typically going to be a BRAF V600E mutation, and this will result in the formation of a benign nevus. And then if a cell in this nevus acquires mutations in other complementary pathways, then that nevus can transform into melanoma. Now, the second route that I just showed you evidence of is for a melanocyte to acquire a more weakly activating mutation in the MAP kinase signaling cascade, resulting in the formation of a field of clonally related daughter cells. These are not going to be perceptible by the naked eye, but they exist. And if one of these cells eventually acquires additional mutations in other pathways and or additional mutations in the MAP kinase pathway, then it too could transform into melanoma. Now, finally, we do think there is a third route to melanoma, as we did see some normal melanocytes with mutations in these other pathways. And that's, as I alluded to earlier, for these mutations to occur in the reverse order. So mutations that affect secondary and tertiary barriers to tumorigenesis might not induce any sort of a phenotype immediately, but this poised cell, when it eventually acquires a mutation in the MAP kinase pathway, which would ignite it, could transform and manifest immediately as a melanoma. We believe this is a common route to melanoma for the nodular subtype of melanoma, which appears suddenly and grows quite rapidly. So I now want to pivot from melanoma to discussing cutaneous squamous cell carcinoma. And cutaneous squamous cell carcinoma is an often overlooked cancer. As an example, they're often not even referred to by their names. They've traditionally been referred to as a form of non-melanoma skin cancer, which has prompted a movement to rename cutaneous squamous cell carcinoma and basal cell carcinomas as keratinocyte cancers. It is not included in cancer registries, and therefore we do not have an accurate estimate of its death toll, but the highest quality estimates range from 2,500 to 15,000 deaths per year in the United States. It's a wide range, but if you take the middle number in this range, that would put its death toll on par with melanoma, gastric cancer, cervical cancer, and liver cancer. So it always has this rap as a non-life-threatening cancer, and I think on a tumor-by-tumor basis, that's true, but it's so common that the absolute death toll is still substantial. Now, cutaneous squamous cell carcinoma was left out of the Cancer Genome Atlas program, and taken together, our understanding of its genomic landscape is poorly understood when you compare it to other cancer subtypes with similar incidence, mortality, and or economic burden. That's not to say we know nothing about its genomic landscape. Shown here are several of the Herculean efforts from various individual laboratories that have attempted to fill this gap in knowledge. And given that these studies exist, it is worth asking, do we even need another cancer gene discovery study for cutaneous squamous cell carcinoma? Now, I would argue the answer is yes. And in support of my position, I want to show a power calculation from Michael Lawrence, Getty Getz, and colleagues. So what they wanted to do here is ask, how many tumors do we need to sequence in order to recognize genes under selection in various cancer subtypes? Now, a gene is under selection in cancer when it has more somatic mutations than the background mutation rate would predict. So essentially, your ability to recognize genes under selection boils down to 2 variables. How many tumors did you sequence? And what is the background mutation rate in your cancer subtype? So we can take melanoma, for example. Melanomas have a background mutation rate of around 15 mutations per megabase. Ignore this data point because this paper was published in 2014. Now there have been nearly 1,000 melanomas sequenced at exome resolution or greater, which would intersect with this blue line, meaning that we are able to recognize genes under selection in melanoma if they're mutated at 5% or more of cancers. Well, what about cutaneous squamous cell carcinoma? Well, unfortunately, its mutation burden is nearly 3 times that of melanoma, and the largest single institution sequencing study only encompasses 40 tumors, and that would put this completely off the chart There's no power curves that intersect with this line, but I would estimate that our statistical power to recognize genes under selection in this cancer subtype is only 50%, meaning that a gene needs to be mutated in 50% of cancers or more. And indeed, from the previous slide, if you look at those 12 or so studies, the only real agreement is not P53 genes that are indeed mutated in 50% or more of this cancer subtype. So to address this issue, we decided, well, let's perform a meta-analysis of these smaller sequencing studies. Towards this goal, we identified studies that made their raw sequencing data publicly available, and we were able to aggregate exome sequencing or genome sequencing data from 105 unique tumors. Unfortunately, this number got cut down to 83 after eliminating low-quality tumors. By far, the most common reason that we eliminated a tumor from downstream analysis was because its sequencing coverage was insufficient after accounting for tumor cellularity. And I cannot emphasize the importance of this enough. I often talk to scientists and they say, oh, but I got 50x sequencing coverage. That's high, right? Superficially, it's high, but if you only have 10% tumor cellularity, that's the equivalent to 5x coverage, which is woefully inadequate. And indeed, cutaneous squamous cell carcinomas are notorious for their prominent immune infiltrate, which often dilutes neoplastic cell content. So it's a very real problem, and I encourage anyone who's carrying out these studies to consider this moving forward. Now, before I show you the data, I think it's a major step forward, but I just want to come out and acknowledge that this sample size is still too low. We have not reached a saturation point where we can confidently say we know most of the cancer genes or most of the genes under selection in this cancer subtype. And so while I have this podium, I want to really encourage the field not to walk away from this problem. We need to keep sequencing at exome and genome resolution cutaneous squamous cell carcinomas. Now, I'm going to briefly overview the number and types of mutations in these tumors. Most of these findings were originally noted were noted in the original studies from which we drew our raw data. There were 5 clinically distinct subtypes of cutaneous squamous cell carcinoma in this meta-analysis. To begin, there was 2 patients with xeroderma pigmentosum. As you could see, and not surprisingly, their tumors had high mutation burdens. And the majority of these mutations were C to T transitions in a dipyrimidine context. This is the traditional mutation that has been attributed to UV radiation-induced DNA damage. Now, interestingly, when we pipe these mutations into a more modern signature analyzer, the Alexandrov analysis that's pioneered by the Sanger Institute, we did not get signature 7, which is the classical or the newer UV radiation mutational signature. We look closely at the trinucleotide content of these mutations, and I'm not going to stand here and say these are not sun-exposed. They almost certainly are, but because these patients are lacking XP proteins, there did seem to be a little wiggle or a little differences, modest differences in the trinucleotide context. And what I think this reflects is some novel biology and the fact that the cosmic mutational data signature database is incomplete. So next, there was patients from the general population. We'll just call these the sporadic tumors. And again, not surprisingly, their tumors had high mutation burdens, and these mutations could, for the most part, be attributed to UV radiation. Then there was mutations from patients who were immunosuppressed. For those of you who are not aware, immunosuppression is a major risk factor for cutaneous squamous cell carcinoma. I believe it increases one's risk nearly 100-fold. These could be subdivided based on the immunosuppressive regimens that these patients were receiving. So most of these patients were organ transplant recipients and were either on azathioprine or some other regimen. Those who received azathioprine, their tumors had high mutation burdens, and as the original authors noted, these mutations could be attributed to SBS32, which has been linked to azathioprine treatment. Those without azathioprine treatment had fewer mutations, and the mutations that did exist were primarily caused by UV radiation. So it seems like azathioprine is really a double-edged sword. It is both immunosuppressive and a potent mutagen, which is really a deadly combination for acquiring cutaneous squamous cell carcinoma, and we should probably phase this out. Then finally, there was tumors from patients with a rare syndrome known as recessive dystrophic epidermolysis bullosa. This is caused by a germline mutation in a collagen gene that causes their skin to be quite brittle. I'm told that literally if they pulled a glove off, it could rip the skin off. And in these chronic sites of inflammation and blistering, it is not uncommon to acquire cutaneous squamous cell carcinomas. As the original authors noted, these tumors tended to have fewer mutations, and the mutations that did exist could be attributed to APOBEC-mediated mutagenesis. So next, we wanted to identify the genes that were under selection in this meta-analysis. And towards that goal, we utilized 4 state-of-the-art cancer gene discovery tools. In total, I believe we nominated here 13 genes, and you could see which genes were nominated by more than one program in the Venn diagram shown here. You could argue this is underwhelming, 13 genes, but this is exactly in line with our statistical power to recognize genes under selection. And so I will acknowledge that this is not a sensitive list, but this is a very specific list. Now, in order to identify genes that were under selection, but perhaps their mutation frequencies were too low to be recognized with one of these discovery tools, we cross-referenced the mutations in this meta-analysis with a cancer hotspot mutational database. The idea being here that if a mutation shows evidence of positive selection from a pan-cancer study, and then we see literally the same mutation in our study, even if it's in just one tumor, there's a strong likelihood that it too is a driver mutation. And this is also a great way to identify gain or change of function mutations in proteins. And so the various hits via this analysis are shown on the right. Now, cancer genes can be activated or inactivated, whether they're oncogene or tumor suppressor, respectively, through other mechanisms in addition to point mutations. And one of those other mechanisms are copy number alterations. So we reanalyzed the raw data and produced copy number estimates from each one of these tumors. We scrutinized the copy number landscape of each tumor and specifically looked for high-level focal amplifications and focal deep deletions. So this is typically going to be a homozygous deletion affecting a tumor suppressor gene. We found focal amplifications affecting cyclin D1, YAP1, and MDM2, as well as a very interesting candidate, RAP1B. This RAP1B amplification, not only was RAP1B amplified, but it also had a mutation. RAP1B is a RAS-like gene, the G12E mutation. For those of you who study RASes, you will immediately recognize this as being analogous to similar gain-of-function mutations in KRAS, HRAS, and NRAS. We also found focal, likely homozygous deletions affecting the CDKN2A, AGIBA, and PTEN tumor suppressor genes. Now, before I provide a general overview of the landscape of driver mutations, I did want to highlight one particularly interesting and novel finding, and that is that we found evidence that a gene called CHUK is a tumor suppressor gene in cutaneous squamous cell carcinoma. So in our study, we identified a total of, I believe, 12 mutations. There were 0 synonymous mutations. I would show them if there were any, but they're not because we did not find any. Most of these mutations are truncating mutations, meaning non-sensor splice site mutations. And then of the missense mutation, most of them are predicted to be damaging. So all in all, as you can see, there really is a skew towards deleterious mutations in this profile. Now, what's not shown here is that nearly every one of these mutations was homozygous or hemizygous, meaning that both alleles were impacted. And this is a very common pattern in tumor suppressor genes. What about the functional evidence? Well, in this case, there was very strong pre-existing functional evidence. And we can start by looking at the original knockout mouse study. So these mice die almost immediately after birth. And that's because they are literally born encapsulated in their skin. So you can see in this image, his arms and legs and tail are literally stuck to its body if you compare him to an age-matched wild-type mouse. Not only that, when you look via immunofluorescence, this is a filaggrin stain. So you can see there's no filaggrin in the CHUK knockout mouse. And this is a stain for differentiation of keratinocytes. So there's no differentiation here, but there is differentiation here. Not only that, this is a general keratinocyte stain. And you could see how much thicker the epidermis is here as opposed to the wild type mouse. Well, what about in humans? Sadly, the exact phenotype has been observed in humans. So there are people who are walking around and talking, and they have one faulty allele of the Chuck gene, and they can conceive children who are born with zero or no functional Chuck alleles, and they have a severe fetal encasement malformation that almost exactly mimics what we see in the mouse. So collectively, there is very strong evidence that Chuck is a key factor governing growth and differentiation of keratinocytes. And in conjunction with our somatic mutational data showing that there's strong selection to lose Chuck in keratinocytes, we believe that this can be credentialed as a bona fide tumor suppressor gene in this cancer subtype. So now I want to wrap up by just providing a general overview of the landscape of driver mutations in cutaneous squamous cell carcinoma. Loss of function mutations in the Notch and P53 pathways were ubiquitous, reaching nearly 80%. This is possibly an absolute just requirement for the formation of these tumors. There were several other signaling pathways that were mutated in the 30 to 40% range, including loss of cell cycle checkpoint control, disruption of critical chromatin remodelers, gain of function mutations in the Hippo pathway, and gain of function mutations. There's really no strong node, but just sprinkled throughout the RTK, RAS, MAP kinase, PI3 kinase signaling cascade. We also performed a statistical analysis to identify mutations that were significantly overlapping or non-overlapping. And after accounting for multiple hypothesis testing, there was only one relationship that remained significant, and that was for loss of function mutations in Casp8. These were enriched in the RDEB tumor, the RDEB subtype. This is likely because Casp8 mediates apoptosis in response to inflammatory cytokines, and these tumors do seem to arise in sites of chronic inflammation. So with that, I want to acknowledge everyone who made this work possible. Obviously, the original authors from these studies, especially Boris Bastian and Robert Judson Torres, who helped me kick off the normal melanocyte work. And then on the keratinocyte work, Darwin Chang, who literally carried out this entire study on his own. And then those who made substantial contributions to the unpublished data, especially, again, Darwin Chang, Bishal Tandikar, and Tuyet Tan, and our collaborators from Northwestern University, Pajaram Jarami and Jeff Sal. With that, I would be happy to field any questions. Thank you. And I'll stop sharing.
Great. Well, thank you so much. It's really amazing the amount of work that you've done and how clearly you're able to present such complicated ideas. And I love all the work in cutaneous SCC. And so it's really exciting to see that. I think we have a couple of questions from the audience. I may turn to those first. Thank you for the great talk. Is there any evidence for epigenetic changes in skin cancer, particularly SCC?
Yeah, there certainly is. I'm not an expert on the ChIP-seq studies, but what we see in both melanoma and squamous cell carcinoma are loss of function mutations primarily affecting the switch-SNF chromatin remodeling complex. Less often, we also see somatic mutations that in EZH2, which provides gain of function to the polycomb repressive complex. And these somatic mutations that affect epigenetic regulators clearly have a functional consequence. And I've been screaming from a mountaintop that we need to be studying this more. So yes, the short answer is yes.
Can I actually just jump in before we get to the second question from the audience with sort of a follow-up question to that? And I apologize if this is unclear and a bit of a theoretical question, but I wonder your general thoughts, someone that's thought a lot about this, about how mutation, the specific mutations in cancer, how that impacts clinical behavior, or does it? Is getting to melanoma the important thing? And from there, other things like the antigens expressing or epigenetic changes or the host really dictate behavior, or are there, predictors of behavior, whether that are genetic, whether in cutaneous SCC or in melanoma?
Yeah, well, as a dermatopathologist, you're well aware that you can almost look at the tumor and predict what the genetic changes are. And so clearly, there is a genotype-phenotype correlation. Now, is there a genotype that causes tumors to be, especially aggressive? Again, I think the answer there is yes, and we need to dissect these mechanisms a little bit more. I'm going to just throw out a hypothesis. I have read some papers that seem to support this, but that also ties into this epigenetic question. When we started seeing these mutations in epigenetic genes, I think we all just tried to latch on to overly simplified explanations, like maybe this is a chromatin remodeler that's, tamping down CDKN2A expression. But I'm not convinced that we found some one-to-one relationship where these epigenetic modifiers are clearly, a node in the MAP kinase pathway or in cell cycle checkpoint control. And so I wonder if the goal from the tumor perspective is maybe to just cause epigenetic chaos, which is just going to cause certain antigens to be up or down. But through Darwinian selection, the ones that get the right cocktail of epigenetic mutations are going to eventually grow out. And I just, it's so confusing to follow the mechanisms that have all been, there's been hundreds of mechanisms proposed, and maybe there are hundreds of mechanisms, but maybe we just haven't latched onto the right one yet. Maybe it's because it's more complex and involves physiology and not just something that we're going to discover in vitro cell autonomous system.
Yeah, thanks for your perspective. I agree fully. Yeah, it's complicated, but thank you for your perspective. And then to the questions from the chat, can you clarify whether UVB or UVA is associated with skin cancer?
Unequivocally, UVB is a driver of skin cancer, and I'm not ready to stand here and say UVA is not a driver of skin cancer. It is clearly more weakly mutagenic. There hasn't really been, in my opinion, a high quality mutational signature attributed to UVA. I mean, I know there's some older studies that show that it induces, some reactive oxygen species that in theory could, but I think when you shine UVA light on cells and look at the mutations before and after, it's a very incremental increase in mutation burden, and they don't really, match up with a known mutational signature. So probably both are bad, but if we're going to go after lowest hanging fruit, UVB is, clearly the worst of the two.
Thanks. I guess I have another question. So what's known about melanocyte homeostasis in skin? So I think this varies a little bit depending on what part of the body you're on. So for example, the palms don't have hair follicles, but for most areas of the body where there are hair follicles, that are thought to contain melanocyte stem cells. Sort of how does that fit into the normal melanocyte data that you presented? And do you think you're capturing those cells? And then realizing this may not fully be delineated, are there interfollicular melanocytes that can also function as stem cells?
Yeah, that's a fantastic question. And in the interest of time, I didn't go into that, but I'm glad that you brought this up. So one particularly surprising finding from our study. So in our talk, I just talked about differences in mutation burden across skin samples. But you can also look at the differences in mutation burden of melanocytes within a skin biopsy. And you see that as a skin sample's mutation increases, not surprisingly, so do the individual skin cells from that sample. But there's this stubborn population of low mutation burden melanocytes that is always present, even in the most damaged and highly mutated skin. So we speculated in our Nature paper that maybe there's some reservoir of melanocytes that are more or less protected from UV radiation, and that as your interfollicular, so this is the melanocytes in between hair follicles, are collecting UV radiation-induced DNA damage. Some are just going to have sky-high mutation burdens, driver mutations, but some are probably going to die in apoptosis. I mean, occasionally a mutation is going to not hit an oncogene, but it'll hit RNA polymerase, and that cell is not going to be able to survive. And we believe that as these cells are dying, that they might be replaced by the reservoir or the privileged niche, such as, for instance, the melanocytes from the hair follicle. Unfortunately, we don't really have spatial information that's retained. So, we just peel off epidermis and sequence what melanocytes come out. I've thought a lot about whether we're getting hair follicles. When you peel off epidermis, I mean, these adnexal structures kind of dip in. It's unclear to me whether those get peeled off with it. So I don't know for sure whether we're sequencing those or not. But it's a provocative hypothesis, and I think that we could probably use some in situ technologies to better understand this. Traditionally, one would probably want to do this in a model organism, but as you and maybe not everyone knows, but mice don't really have true interfollicular melanocytes. Most of their melanocytes are in the dermis, and so it's not really in my opinion, very well worked out, the homeostatic mechanisms operating on human melanocytes. So this is a major gap in knowledge we need to address.
Thanks. Yeah, thank you. Still a lot to learn. A couple other questions from the audience. In terms of skin regeneration, skin cancer overlap of genes, and wound healing overlap of genes. And there's a question about cancer overlap. I guess maybe to rephrase if I'm understanding it correctly, in terms of the signatures that you're seeing, is there an overlap in, clearly tumor promoting genes, and, perhaps with the question of the EV patients versus wound healing? And I think you mentioned a little bit about how certain genes are, sort of enriched in the RDEV patients. Do you have any additional comments?
Yeah, so, the original wound healing signature that didn't actually pan out. So as my understanding is that bulk tumors were sequenced, and if you just add media to fibroblasts and they upregulate genes, and you see the same genes upregulated in bulk tumors, and so it was postulated that these must be emanating from the fibroblasts. But when you do in situ studies, if you add media to fibroblasts, they're happy and they grow, and you see proliferation genes. And if you see cancers, they grow fast, and so you see a lot of proliferation genes. That's not to say necessarily that the microenvironment's not important in playing a role. And I think that's something that we need to continue studying. These studies have been that I just presented are maybe a little bit myopic, and they focus on one dimension, genetics, and one cell type, which is the tumor or potentially future tumor cells, which in the case of normal melanocytes. But obviously the microenvironment, especially the immune system, are playing a role, and that's reflected in even though it was a one-dimensional study, we do see these caspase mutations that are clearly selected for in tumors with high levels of immune infiltrate. And I'll also cite some work by Gady Getz. They did one of these massive pan-tumor analyses, and this was totally agnostic of cancer subtype, but they said, in tumors with high immune infiltrates, what are the somatic mutations that are most common or enriched? And Cas8 was also in that study, as well as beta-2 microglobulin. So obviously the microenvironment can cause selective pressures to occur in the tumor cells. But it's a good question. We need to research this more.
Thanks. And then one last question from the audience that I think is related to the immune response against skin cancers is, do you have any thoughts about the potential effects / mechanisms of JAK inhibitors in skin malignancy?
Yeah, again, I'm getting outside my lane here a little bit, and the complexities of the crosstalk signaling between immune cells and a pathway I don't know a ton about, so I'm not gonna make fool of myself and go down that, but I'll just defer to the immunologists and the experts in the room that we need to learn more.
Agreed. Yeah, no, I think we do need to learn more. Well, great. I think that's it for the questions. Just pause for a second, just give anyone else a chance if they'd like to ask a question.
I just had one actually. There was a slide that you showed about the burden of mutations in the patients, and I was just wondering, was there any variation in kind of burden of mutations in ethnic skin, if that was looked at?
Yes, and there was recurring answer. We need to learn more, but we did sequence melanocytes from a subset of patients who identify as being of African ancestry, Hispanic, or of Asian ancestry. And generally speaking, they had fewer mutations. And that's maybe obvious, expected, but there have been some people that have postulated that, the tumor suppressive mechanisms are not so obvious, and I can't rule that out. But I don't know. I'm tempted to not overthink this and say that, pigmentation protects our skin from sunlight, which protects our skin cells from accumulating mutations, and that's probably why people of European ancestry are more likely to get skin cancer. But maybe it is more complex, and it involves reactive oxygen species, and a lot of different mechanisms have been put forward. But very, very interesting area that we want to keep focusing on.
I actually have a quick question. Have you looked at acral melanomas yet?
We looked at a very small number of melanocytes from acral skin. They had extremely low mutation burdens, as would be expected. But what I want to do is I want to look at more, and in particular, you might be aware of this, but acral melanomas, they have this very distinct and unique mutational signature. It's called typhonas. I think it's only ever been observed in one other cancer subtype. And so, my question is, do normal melanocytes, pre-neoplastic melanocytes, is there some baseline typhona activity occurring? Or is this something that, a phenotype that's acquired only in the cells that eventually become tumors? And, if this is something that's operating at baseline, is there some way that we could develop some therapeutics to, stop it from operating? Clearly the etiology and route to cancer for acral melanoma is very different. And, I think this is, we've got a system in place to maybe answer some key questions there.
Great. Well, it looks like it's 1 o'clock. So thank you so much for sharing your expertise with us. This is really just a tremendous talk. And thanks everyone for tuning in today.
All right. Thanks, guys.
Bye-bye.